Hi guys, welcome to another new video from our channel Bioorganic Hub. So as you can understand in this video, as you see here, we will discuss the fermentation. So what is the definition of fermentation? What is the scientific basis? And when the fermentation happens, that's the most important question. When you need fermentation, whether it's only need for yeast, the lower eukaryotic cell, or also you also need for a higher eukaryotic cells. So that we will see. And one thing to clear, the fermentation is not only required for the yeast, that is the lower eukaryote, it also required for the higher eukaryote like animal, but in certain specific conditions. Okay, that we will see in this video. Okay, so let's start. First, to start the fermentation and what is fermentation, I would like to start like that. The food you are taking, the food source you are taking from external of the body, that you are taking glucose. You are taking glucose from that food source. Now you have to survive, you need energy, and that energy will be produced in your body as a form of ATP. So adenosine triphosphate, you know, that's the source of energy in your body for any metabolic or any other physiological process what you are going in your body. You need ATP. So what happens, you need ATP from the food source. So the food source to ATP conversion, from the food source you are taking from outside to ATP conversions, is going through 4 steps mainly in the eukaryote, like animals, us, a human. Okay, so in this type of process there are mainly 4 steps. One is the glycolysis, and the next step is the Krebs cycle, and then 2 other steps, electron transport chain, and etc, etc, that we will discuss in another video. But the main focus of this video is that after glycolysis what happens for the both lower eukaryote and higher eukaryote. So you can understand, after the glycolysis what happens. Glycolysis means the glucose to pyruvic acid conversions. When glycolysis is happening, this is just a breaking down of your glucose molecule. You have the glucose, that is 6 carbon containing cyclic organic molecules, and you have to break down this glucose to pyruvic acid. Okay, two number of pyruvic acid forms from one glucose molecule in glycolysis forms 2 moles of pyruvic acid. Okay, so the point is that after the formation of pyruvic acid, what will happen? Okay, and it will go to Krebs cycle. But before the pyruvic acid formations, that is the glycolysis process, that doesn't need any oxidation, or that doesn't need any oxygen. But after the glycolysis, that is the Krebs cycle, electron transport chain, that's all. So Krebs cycle needs oxygen. So whenever you have oxygen, like I have said, the many eukaryotes like human aerobes. Actually human is called aerobes. That means they survive only in presence of oxygen. So why you can survive only in presence of oxygen? Because you need energy to survive, that is ATP, and this ATP will form by a large complex series of reactions, sequence of reaction, and in this sequence of reaction one important step is Krebs cycle or citric acid cycle, and in this citric acid cycle you need oxygen. If you don't have oxygen, you can't survive. You can do that steps, that's why you can produce ATP, that's why you don't have any energy, though you are taking your food source. So that's why human can only survive in presence of oxygen. Human are aerobic animals, that's why they are said human are aerobes, that can only grow in presence of oxygen. That means to convert food source to ATP you need oxygen in the Krebs cycle. But whether this possible in absence of oxygen? So the question is, in absence of oxygen, what is the fate of this pyruvic acid, what has been formed in the glycolysis? That is the answer of fermentation. That is the answer, the fermentation. So whether this fermentation happens for animal cell in special condition or any other cells, that we will see. That some eukaryotes like yeast also can grow either in presence of or in absence of oxygen. That means they have some either way pathway, that's a managed pathway, they don't need oxygen in that pathway. Okay, that's the procedure for yeast, lower eukaryote. So they realize ATP, they also need energy to survive. All the animals need energy to survive. So they realize ATP produced by fermentations, that is called fermentation. That is the productions of ATP or the fate the without oxygen fate of pyruvic acid is actually the process of fermentations. So fermentation is the process by which in yeast pyruvic acid without oxygen, in absence of oxygen, converts to some other molecules, and we will see. And in humans there are some specialized condition in which after glycolysis pyruvic acid will form, and there are some specialized condition where this pyruvic acid will be converted to some other organic molecule. So this is the anaerobic catabolism of the glucose. So anaerobic catabolism of the glucose is called fermentation. Fermentation is nothing but the anaerobic catabolism of glucose. Fermentation is nothing but the anaerobic catabolism of glucose without oxygen. They don't need oxygen. Catabolism means breaking down of biomolecules to its small portion of small organic molecule. Okay, like pyruvic acid, small molecules, lactic acid, small molecules, but you have a glucose, a big ring structure. That is catabolism. Without oxygen, in absence of oxygen, catabolism of glucose, that is the main food source, is called the fermentations. Yeah, so fermentations, what happens in fermentations? Anaerobic catabolism of glucose, that is glucose molecule will be converted to pyruvic acid. So after that pyruvic acid will be converted to some small molecules in absence of oxygen. That is called the fermentations, and that you will see in the next slide. Let's first discuss about yeast. We have said the yeast normally does by fermentation. They survive in absence of oxygen. So they take some other pathway to form ATP, to produce ATP in absence of oxygen. Okay, so if you go through the steps, for a yeast anaerobic metabolism, this is called the anaerobic metabolism. So you have first glucose, that is you have taken from another food source, and in glycolysis this is converted to pyruvic acid. So in glycolysis plus acid has been formed from the glucose. And in this process, you know, ADP is converted to ATP and NAD plus. NAD plus is NAD plus. I have forgotten to write NAD plus. NAD is converted to NADH. That is reductions of NAD happens is converted to NADH. And ADP will convert it to ATP. So remember, in glycolysis, you can see in another video of my lecture what is the glycolysis and what is the mechanisms of all about things. So in glycolysis, what happens? Pyruvic acid will be formed and concomitantly 2 ATP molecule will be produced. Now what happens after pyruvic acid formations? After glycolysis, you can see there are 2 double bond O. Okay, so this hydrogen will be attached with this oxygen, and this oxygen and oxygen will be exhaled by the system as a carbon dioxide. So carbon dioxide will be produced by a decarboxylation procedure. This is called the decarboxylation, means carbon, oxygen, oxygen. It will form a CO2. This is called carbon dioxide, and this is released. That's why called decarboxylation. So by some decarboxylation procedure, it releases CO2 and form acetaldehyde. And then what happens? Then just reductions. So you have to remember, for yeast anaerobic metabolism, pyruvic acid, what forms after the glycolysis, this converted to the ethanol. By the concomitant reduction of NADH. Concomitant oxidations of NADH. You can understand, whenever oxidation will happen, concomitantly reduction will happen. You have acetaldehyde will be converted to ethanol. That is the reduction. This is a simple reduction, you can understand, because you have CHO. This is a oxygen, and you are putting hydrogen in this carbon atom. That's why this is called reduction. When hydrogen addition will happen, this is reduction. So acetaldehyde is converted to ethanol. That's why it is reduction. And concomitantly oxidation have to happen. Oxidation happens, NADH. This NADH has been formed from glycolysis. In the previous stage, glycolysis resulted formations of NADH. That's the reduced form of NAD. Now this NADH will be oxidized again back to NAD plus. Okay, by the yeast anaerobic metabolism, that is called the fermentation. So this procedure, pyruvic acid to ethanol conversion, that is called the fermentations. So this fermentation, one important step is that fermentation is very important to survive the yeast. Why? Because in fermentation you are forming ethanol. That's not an important point, but important point is that you are forming NAD+. This NAD+ is very important in a intermediate step of glycolysis. You can understand here NAD+ is converted to NADH. So NAD+ if you don't supply, if you can't supply in the term of glycolysis, then you can't manage the glycolysis cycle. Okay, so to accomplish glycolysis, to continuation of the glycolysis, you need NAD+, but from where you will get the pool of NAD+? You will get the pool of NAD+ just after the glycolysis, called the fermentation process. So fermentation is not only important for the pyruvic acid to ethanol conversion, but it is mostly important to the filling up the pool of NAD+. And this NAD+ will be again reused by the glycolysis sequence of reaction to be reduced as NADH. And this NADH, see, this NADH again will be used in the fermentation process. So there is a positive feedback loop. NADH converted to NAD+, NAD+ going to glycolysis, and glycolysis again forming NADH, NADH coming to fermentation again. So there is a positive feedback loop happening in the fermentation process. So glycolysis and fermentation go very positive feedback loop. Okay, that's how yeast forms ethanol in absence of oxygen, and that's called the fermentation process. Now what will happen for the animal cell? What will happen for the fermentation of animal cell? That we will see in the next slide. Okay, so what will happen for the animal cell? What will happen for the animal cell anaerobic metabolism? And one thing I want to say here in this point, that when animal cell needs the anaerobic metabolism, that is called fermentation. When, let's say, you don't have oxygen for the animal cell, then although you don't have oxygen, it can form ATP, and that will form by the fermentation process, that is anaerobic metabolism, and there lactic acid actually formed. Lactic acid formed in absence of oxygen. But you can understand for the yeast, just let me take the laser point, yeah. But you can understand for the yeast, it has been changed to another organic molecules, that is actually you have seen in the previous, that is actually ethanol. But in the case of animal muscle cell, you have converted to lactic acid, for pyruvic acid to lactic acid conversion. So in animal muscle cell, pyruvic acid to lactic acid conversions, or the formations of lactic acid in absence of oxygen, that is called the fermentation. Now the question is when it's required, when the process is required. You can say like that, whenever you are doing exercise, have you ever thought why you are feeling pain after exercise, after a long exercise, not a short, after a long exercise you will feel pain? Why? That's the point, the fermentations. That's the science. Fermentation resides here. So whenever you are contracting your muscle, okay, let's say you are contracting your muscle for a long time, so there are shortage of oxygen in the animal tissue. So whenever you are making exercise for a long time, for a prolonging time, you are contracting your muscle, there will be shortage of oxygen. There will be scarce of oxygen in your animal tissue, in your muscle tissue. Now what will happen? First step, that is the glycolysis. You have taken your food before exercise, so that food will be converted to pyruvic acid by glycolysis pathway. But glycolysis, remember, don't need oxygen. You can see in another video of my glycolysis lecture, glycolysis doesn't need any oxygen. So glycolysis has been happened in absence of oxygen, but you have less amount of oxygen in your animal tissue, muscle tissue. Then the next point, that the Krebs cycle or citric acid cycle, that will not happen. So what will happen? In that condition, anaerobic metabolism will happen. And what will happen in anaerobic metabolism? This NADH, the same concept as like the yeast. NADH you have formed in the glycolysis, and that will be converted, that will be oxidized to NAD+, and that will make a positive feedback loop for the glycolysis. That is this NAD+, this NAD+ will supply the pool for the NAD+ required in the glycolysis sequence of reactions. So by this oxidations, concomitantly pyruvic acid will be reduced to which molecules? This will be reduced to lactic acid. This will call lactic acid. See, this is just a reduction. COOH has been kept as intact. COOH and COOH has kept intact. This CO bond has been reduced to CH2OH. So hydrogen addition is reductions. So reductions has happened and this will form lactic acid. Now what happens? See, when you were making a prolonged exercise, so what will happen? This lactic acid is forming in your animal tissue. Then it will comes to blood. Now if it comes to blood, okay, so lactic acid has been from cell comes to blood. Now this blood, if accumulates for a long time, it can make pain, because it will be accumulated in the muscle tissue. These accumulations of the lactic acid in the muscle tissue actually makes a pain after the prolonged exercise. Have you ever thought it? This is also science. Okay, so the accumulations of lactic acid has made a pain after your exercise. Okay, so the take home message is that animal muscle cell anaerobic metabolism in a special condition forms pyruvic acid to lactic acid conversions. But in yeast, you have seen pyruvic acid has been converted to ethanol. So the final sequence of reactions, if we want to see, then what will happen? Overall reactions of anaerobic metabolism with glucose to ethanol conversion. Mind it, glucose to ethanol conversion is not going to on a single step. First glucose to pyruvate, that is called glycolysis, and then pyruvate to ethanol conversion. That is for yeast. But in the animal muscle cell, the main difference is glucose will be converted to pyruvate. Up to that, this is fine. But after that, after the pyruvic acid formation, what will happen? After pyruvic acid formation, it will converted to lactate in animal muscle cell. But in yeast, it will be converted to ethanol. That is the main difference for the yeast anaerobic metabolism and animal muscle cell anaerobic metabolism. Ethanol is formed in yeast and lactate is formed in animal muscle cell, and other is same. You have ATP formations, you also hear ATP formations. That is the survival, that is the energy source you have formed by the fermentation. So in both the case, fermentation will produce some ATP which acts as a source of energy, but here ethanol is forming and here lactate is forming. And remember, fermentation can be many types, like batch fermentation and any other continuous fermentation. That I have another video. You have seen another video about batch fermentation, continuous fermentation, what people use in industry, because fermentation is the basis principle of the productions of beer and wine. So the beer wine production depends upon the fermentation mechanism. And remember one thing, the case was in that case for the yeast, the decarboxylation. That's why you have a CO2 molecule here, CO2 productions here. But the animal muscle cell doesn't go by the decarboxylation stage. That's why it doesn't produce any muscle cell. So that's about the fermentation system. Okay, that's all about fermentation. Hope you understand all the concept. Okay. Hope you understand all the concept, and then thank you.